Hi, everybody. Thanks so much for joining us for another Molecular Devices webinar. Today we're going to be going through, simple and flexible high-content imaging, enabling quantitation of complex biological events. The main speaker is going to be Vicky, which I'll introduce in a moment. But I'd like to visually go through some of the items that Leah just mentioned. So if there's any issues, technical or otherwise, there's a 1-800 number, and more appropriate might be to send us a Q&A. The way to send us a question is to press on the Q&A button at the top of the toolbar to expand the Q&A window. When that window pops up, type in a question here. Choose all panelists so that Vicky, Leah, and myself can see the question. And then press the send button, and we'll see that question and try to respond appropriately. Please send questions about the application anytime during the webinar, and if you have any technical issues, please do so now. So I'm the host. I'm the product manager for the cellular imaging here at Molecular Devices, and I will do a little introduction on HCS in general, and when Vicky is done, I will talk a little bit about the new MetaXpress Micro XLS system. Vicky will be going through how to set up simple and flexible tools to quantitate complex biological events. Why are we having this webinar and developing products here? It takes billions of dollars and many years to bring a drug candidate to market. And the later in development time, this is time in development, the more expensive that drug is to bring to market. So discovery is the shortest time, preclinical, and as you go on in time, the price for that drug, bringing that drug to market increases. And many drugs fail, and the number one failure mode is animal toxicity, and cardiovascular and liver toxicity are the most common organs affected. And this can be early in the drug development process, or actually later, and drug recalls, the same systems are affected. And only 5% of drug candidates make the market. So we strive to bring products to market that help identify better candidates early, so that you can find those molecules that are going to be efficacious and not toxic early in the discovery process. So Vicky's going to look at through two imaging processes: one cell signaling through GPCRs, and the other looking at mitochondrial toxicity that will help discover those molecules earlier rather than letting the molecules progress through development and easily pulled off late in the discovery process. The HCS workflow for those unfamiliar with high-content imaging is to capture cells, put them into multi-well plates, acquire images, identify objects doing the image analysis, and that's where the webinar is going to concentrate today. Obtain multi-parametric data output and make sense of the numbers to hit find and data mine the large number of parameters and conditions that were screened. What does Molecular Devices have for high-content imaging? We have a wide field system, the MetaXpress microsystems. I'll be talking about that at the end after Vicky's section. We have a confocal screening system, the MetaXpress Ultra. We can analyze third-party images and bring those images into our data management solution. Everything is tied together by MDC Store, which is our data management solution. Vicky is going to be talking about some image analysis capability within MetaXpress, and we have the MetaXpress protocol option to speed up analysis. That takes these images that are collected and crunches those images into numbers. And we have Acuity Express to make sense of all the numbers that are obtained. Sorry, so I'll just progress through those things. That's the Molder HCS system. And at this time, I will turn it over to Vicki to talk a little bit about MetaXpress and setting up analysis in MetaXpress.
Thank you so much, Grisha. Can everyone hear me okay? So thank you again for joining us today. As Grisha very nicely illustrated, the high-content screening approach can add value to your drug discovery campaign, enhancing capability to eliminate toxic or non-specific compounds very early in the drug discovery process, reducing attrition later on when it's much more expensive. In order to maximize the value from your high-content assays, Molecular Devices offers a comprehensive package of image analysis tools spanning turnkey application modules, which are plug-and-play solutions for analyzing images from our most common assays, to our very powerful journaling capability, which provides unrivaled flexibility for image analysis. Bridging those capabilities are our newest addition to the product line, which are the capability to build your own custom modules, allowing you the ease of use of application modules while harnessing the power of our more complex analysis tools. So what we're going to focus on today are two custom modules that highlight some assays that are fairly common in the field, GPCR internalization and mitochondrial toxicity. Before we walk through those individual modules, however, I'd like to just give you a quick overview of our custom module editor interface, where in 3 easy steps we can extract our data from our images of interest. So in this particular example here, we have a nuclear punta assay. What we're doing here is we're defining our objects to measure, our nuclei, and then our puncta region. We're choosing up to 50 data outputs to extract from those images. And then once we've generated that module, we can save it and run it just like a pre-configured application module. So for those of you that are familiar with our plug-and-play application modules, the workflow is very similar once you've created your module. So to look at in a bit more detail at the custom module editor, we'll be working live with the software in a few minutes, but just to give you a quick orientation and point out some of the nice tools that we've built into this interface. This is the general interface. Your images that you've chosen will appear in the center of the window. We'll have a complete panel of tools here at the top that contain everything you need to segment your images and extract the data that you're looking for from those images. Once you've selected a tool, in this case, we've selected a tool that will allow us to find our nuclei in the interest of finding our nuclear puncta, using a very nice feature called the Click to Find tool. So when we activate this tool, we can click on individual nuclei directly on the image till we capture a representative population of nuclei. As we click through, the values that are affiliated with those nuclei will populate automatically in the step card. So we have a range of sizes and intensities that are being recorded as we click. When we click apply, we can see that when those settings are applied, the result mask that appears here on the right-hand side is showing us how the software is picking up our nuclei. So you can see it's doing a very nice job. We can perform a similar step for our puncta image in this case. And we can see here when we select when we review our final measurement mask that we've picked out our nuclei in the cyan mask and our nuclear puncta here in the yellow. You can see that all non-nuclear material is being excluded from this analysis. So this is a very nice approach that you can use for picking out any type of nuclear foci. So you can imagine several applications for this. Once you've generated your final measurement mask, you can select your measurements of interest and then apply the settings to review those measurements interactively with your images. So in this case, we've selected a line of data here in the data table on the right, and we can see that the cell that's affiliated with that data point is being highlighted in orange on the image itself. We can select a range of nuclei and have the reciprocal happen, those lines in the data table that are affiliated with those nuclei will be highlighted. So this is a really nice feature that allows us to interactively review our data and optimize our settings accordingly. Once our settings are to our satisfaction, we can save our module with any name that we like. And once we click Save, that module is inserted directly into the MDT Store database and is immediately available for analyzing everything from a single site to a stack of placeholder data. Once those data are generated, we can review those numerical outputs in a user-configurable heat map. This is a very nice feature of our software that allows us to visually QC our data before we perform any downstream analyses. So the first custom module that I'd like to review is the GPCR internalization custom module. So for those of you that are familiar with our Transfluor technology, it takes advantage of the fact that when a G-protein-coupled receptor of interest is stimulated by its ligand, it is then phosphorylated and internalized, which is part of the desensitization process. We take advantage of this generic phenomenon with GPCRs by tagging beta-arrestin with GFP. So beta-arrestin is recruited during the formation of pits or vesicles as that desensitization process proceeds. And as you can see in this live cell images that were captured on the MetaXpress micro, that we can visualize the relocalization of that beta-arrestin GFP as the stimulation with the ligand of interest proceeds. This is a very powerful generic platform for looking at multiple GPCRs. This slide just highlights a cross-section in the literature of GPCRs that translocate with beta-arrestin, and we've again taken advantage of that platform by tagging beta-arrestin with GFP. So in the interest of generating a very specific read around tracking the internalization of our GPCRs of interest, we've highlighted this particular example where in this image we're showing U2OS cells that are co-expressing our beta-arrestin GFP and the beta-adrenergic receptor. This particular sample has been treated with a dose of ligand that causes the receptor to internalize in these green pits that we see here in the GFP, and we've counterseen the cells with us to visualize the nuclei. This image set was generated on an MetaXpress micro with a 20x lens, and the analysis that I'm about to show you was generated in MetaXpress version 5.2 with our custom module editor. So the goals of this particular analysis are, first of all, to limit detection of our vesicles or pits to the cytoplasm of our cells without having to incorporate a whole cell dye. So our standard Transfluor application module is very powerful. It does offer the ability to count our pits or vesicles, which are here highlighted in the dark blue mask, as well as our nuclei. However, it does not have the flexibility of looking at things like cell shape or defining cell boundaries. So this is actually building on the power of that Transfluor module and giving us a broader array of measurements available, as well as allowing us to specifically exclude extracellular material, which may be non-specific. So we can, as we'll see when we walk through the module, we'll have the flexibility to also make other measurements that may be useful to us in monitoring the effects of our treatments, such as looking at cell area, morphology, or other phenotypic measurements. We can also, of course, monitor the effect of our compounds or treatments on cell count by counting the number of nuclei or the number of cells. So with this, I'm going to exit to the software so that we can work with this custom module live. For those of you that are familiar with MetaXpress, this is our primary interface, where in our review plate data interface, we can actually highlight a subset of wells, and we'll see up here a montage that allows us to review the effects of our treatments visually before we perform any downstream analysis. As we click through the thumbnail images, we'll see that our high-resolution images will update automatically, again allowing us to review the effects of our treatments. To create a custom module or to modify an existing custom module, we simply launch our custom module editor from this review plate data interface. And we'll see I've pulled up our module already with a representative well where we see internalization of our receptor. So as you can see, the images appear here in the center of the screen. We can scale those images to display the pixels that are most meaningful to us, so we can make it brighter or dimmer. We have a panel of tools here at the top that, again, contain everything we need to build our analysis. In this case, it's going to be a combination of a total cell indicator as well as our Transfluor measurement, our pits and vesicles. So the first step to generating our module is to actually drop in an application module object. So you'll see when I expand this menu that I have access to all of the segmentation masks that are affiliated with any application module that I have license for. This provides a very powerful starting point for an analysis like this, and in this case, we're going to select our Transfluor object that will allow us to identify our nuclei as well as our pits and or vesicles. So for those of you that are familiar with our standard application modules, you notice that the interface is very similar. In the step card, we see that we can choose a source image. In this case, for our pits and vesicles, it will be the 60 image. And we have three simple inputs that we need to select our objects of interest, in this case, our pits. Similarly, for our nuclei, we have the ability to select, in this case, our DAPI image. And again, we can populate this information here to select our nuclei. As we pointed out in the introductory slides, we do have a very nice tool called the Click to Find tool that can allow us to interactively select a subset of nuclei. So if we zoom in here using our scroll wheel, we can then choose some nuclei. They will appear again in the panel on the right, our result images. And you'll see that the numerical values in this panel are updating as we click through. Once we've chosen a representative population of our nuclei, we can then click Apply to review the effect of our settings. We can also rename our result images to anything that suits us. So we could say we want beta 2 adrenergic receptor pits to make it a bit more specific, for instance. The next step is to actually now define our cell boundaries. And to do this, we're actually going to use another application module object drop-in. So from this same menu, we're going to choose our cell scoring object. And what this is going to allow us to do is to take advantage of the background staging or the background haze of our GSP tag data arrestin. So if we make this image a little bit brighter, you can see that we have a haze in the background that we can actually use to define the cell boundaries. So this is what we're going to take advantage of so that we can define our cells without incorporating any extra steps in the staining. So using a similar approach, we can select both our nuclei and our cells, or we can simply just populate the nuclear information from the previous step card. And when we click Apply, we'll see that we've got all of our nuclei defined, as well as our positive cytoplasm here on the right-hand side. If we switch to the split view, we can overlay all of our masks with our source images and interactively review how those masks overlay with our source images. So we can ensure that we're picking up the cytoplasm in the way that we would like to. Now, our next step is to actually generate a whole cell mask, because we want to have the capability to make measurements on cytoplasmic objects as well as make measurements on our nuclei themselves. So to do this, we're going to perform what's called a logical operation. This sits in the Modify Objects menu, and what this step does is it allows us to use some simple logic to combine one binary mask with another, in this case our nuclear mask with our cytoplasmic mask. And in this case, we want to keep all of the objects that are covered by each mask. So we're going to use an OR argument to do that. And you'll see when we switch back to the side-by-side view that we have our two source masks and then our whole cell mask. So that's the result that we want to see here. Finally, we want to be able to make a specific measurement of the pits in the cytoplasm. So what we're going to do here is we're going to perform what's called a Keep Marked Object step. This also sits in our Modify Objects menu, which in general contains tools that allow us to combine multiple masks with each other or make modifications to those masks. So in this case, we're going to use our beta-2 adrenergic receptor pits as one of our source images, and we want to keep only the pits that are affiliated with our positive cytoplasm. So we can see that if we switch back to the split view, that we're going to be excluding any pits, in this case, that are affiliated with our nuclei or that are overlapping our nuclei. Now our final step is to define our measurements. So in this step, what we want to do is first create create an object to measure map. This is going to allow us to define the outermost boundaries of the objects that we want to measure. In this case, it's going to be our whole cell mask. and we're going to make measurements in the 50 channel. If we extend by clicking on the ellipsis button, we can see that we have access to up to 50 measurements that we can make of our cells. So in this case, we've chosen to make some general measurements of area, shape factor, length, and breadth. So these are phenotypic type measurements. We can also look at the intensity of our GFC across the entire cell. So we've chosen to do that here. We can make similar measurements on our nuclei. So any objects that are defined in these subsequent objects to measure fields are going to be objects within this primary mass, so within this whole cell mass. And we've chosen to make measurements on all of our pits, our nuclei, and we can also add a step to look at our cytoplasmic pits. Let's do a simple count. We can name these outputs whatever we like, so we can custom define the module to our liking. So this will be cytoplasmic pits per cell. Apologize here. Okay, so now we've defined our measurements. We can now apply those measurements and review the final measurement mask. So we can see here that our cells are outlined in cyan, our nuclei are in yellow, all of our cytoplasmic puncta are in pink, and our nuclear or anything that's not defined as cytoplasmic is in dark blue. And again, as we reviewed in the earlier slides, we can review our data table interactively. We can highlight some cells of interest. If we zoom out here, we can see that these cells here are key to these data points. Similarly, we can click on any cells of interest within the image and review the data points that are affiliated with them. Another nice feature of our custom module editor, so if you see a measurement that you'd like to optimize further, you can actually jump back in the module just by clicking on one of the step images in the film strip here at the bottom. So say we want to make a modification to our total cell mass, we can double click on the film strip for that particular step and jump right to that step. Here. So once we've generated our measurements and we've reviewed them, we've optimized our settings to our liking, we can then save our custom module with any name that we like. We can also modify an existing custom module and save it with a different setting name. So if you're changing cell lines, if you've seen changes in your dyes, if there's anything different about your biology, you can make modifications to an existing module and save it. And it will again update automatically in the database as soon as you save it, and allow you to then run it on your image sets. Another nice feature that I like to point out here is our export PowerPoint features. If you're generating an analysis for which you would like to provide a walkthrough for your colleagues, for instance, you can export any visualization in the software directly to PowerPoint. It's a very nice feature of the software. So once we've generated our Transfluor custom module, we'll go back to our slides here, and I can show you a bit of the results. So as I mentioned, we do offer a very powerful standard application module that allows us to make these types of measurements. In this case, pits per cell is one of the measurements that people often track when monitoring effects of a compound on GPCR internalization. So we can still capture that measurement here with our custom module. However, we can also incorporate other measurements that are going to allow us for a more data-rich output from this particular assay with minimal effort to generate those measurements. So here we have our cytoplasmic pits per cell measurement. We can see that it also trends similarly with our increasing ligand concentration as we move up the column. We can also see that a change in the whole cell GFC intensity, as well as a change in shape factor, while we're not seeing any real effect on our cell density. So this particular ligand is not impacting the health of the cell significantly. So this is just a flavor of the types of measurements that we can extract from a custom module that's based on two standard application modules. In this case, our cell scoring module and our Transfluor module. So we're also, not only are we ensuring that all of our kits that are measured are intracellular, we can make an assessment of our treatments on all of these whole cell measurements for richer phenotypic readouts. Our next custom module that we'd like to highlight is our mitochondrial cell health custom module. So this allows us to make measurements on, to measure the effects of compounds on mitochondrial health. Just to quickly review mitochondrial structure and function, as we all know, mitochondria are the powerhouses of our cells and are key players in many important biological processes. The ones that we're going to highlight today are general mitochondrial function as well as a potential role in apoptosis. Several of our customers are interested in looking at mitochondrial morphology, and we can see from this illustration from the literature, when cells are exposed to a toxic treatment such as this ethanol treatment, we can sometimes see a change in morphology. So in this particular example, the mitochondrial structure is going from a more fibrous morphology to a round morphology. So this is something that we're interested in tracking as a potential read of toxicity. In addition, there's also evidence in the literature that in certain cell lines, mitochondria will migrate to the peri-nuclear region in response to an apoptotic stimulus. So this is another measurement that we're looking to build into our mitochondrial health module for this reason. So as we saw very nicely when we were working in the software with the Transfluor example, we can review all of our image data in a montage. So for this particular assay, we have a dose response of compounds going from low concentration to high, from right to left, as we proceed across the plate. And in this particular example, each row is a singleton of a concentration response of each compound. So there's a panel of compounds here that are being profiled, and you can see an effect, a toxic effect on the cell member just by looking visually at these images, as well as a decrease in the mitochondrial red stain. So in this particular example, we're looking to find our best way to assess the effects of our compounds on mitochondrial health. These images are taken of fibroblast cells, which are stained with mitochondrial red and counterstained with us to visualize the nuclei. The images were generated on an MetaXpress Micro XL with a 20x lens, and the analysis again is run with MetaXpress 5.2, featuring our custom module editor. So the goals for this particular analysis are to quantify both the average and integrated intensity of our mitotracker stain. This is an indication of mitochondrial health. To classify and measure our fibrous mitochondria versus the fragmented mitochondria, so healthy mitochondrial networks in our fibroblasts versus those that are undergoing fission in response to a toxic stimulus. We're also looking to measure the intensity of mitochondrial red in the peri-nuclear region. So this is a phenotype that's potentially related to the induction of cell death, as we saw in the example from the literature in the prior slide. And of course, we want to monitor general toxicity by counting, by monitoring cell loss across our treatment course. So this is just a closer look at the result mass that we're looking to generate. In this particular example, our nuclei are highlighted in purple. The cytoplasm of our cells is in teal. Our fibrous mitochondria are in pink. Our fragmented mitochondria are in blue. And then we've also highlighted the subpopulation of mitochondria, both fragmented and fibrous, that are coincident with a peri-nuclear ring here in the white. So a little bit more complex than what we were working with previously. So again, we're going to now work live with the software to show you how we built this module. And I've already pulled in some images here for us to review. Let's go back to the segmentation task. So again, our mitochondrial stain is here in the orange, and our hook stain is in the blue. So similar to our approach for defining our cell boundaries for the modified Transfluor read, we're going to use an application module object called cell scoring object. And in this case, we're going to define our nuclei again by the DAPI stain, and then use our Psi3 channel, our mitochondrial red, in the cytoplasm to define our cell boundaries. And when we click apply and look side by side, we can see that we're picking up all of our cells. So all of the nuclei being classified as positive, and then our cytoplasm is really nicely defined here. So I'm going to take a quick look at that mask on its own. We can actually turn off some of these result images, allowing us to focus on the steps that we're most interested in. So again, we can compare here the cytoplasmic stain with our background of our mitochondria, which is what we're using to define those regions. And we can look interactively in the split view. So once we're happy with our whole cell mask, we can perform, again, a logical operation to generate a whole cell mask, adding our positive cytoplasm that we've generated to our positive nuclei mask that we've generated in our cell scoring objects drop-in. So again, we'll use, from the Modify Objects menu, a logical operation step, where with an OR argument, we're going to combine our positive cytoplasm with our positive nuclei to generate a whole cell mask. And we can see when we look at the side-by-side view that we've combined these perfectly. Our next step is to define our mitochondria. So in this particular case, we notice that while the hazy background that we see with the mitochondrial stain is actually a benefit, allowing us to generate a whole cell mask, again, without incorporating an additional whole cell dye, it's advantageous to us to be able to better define our mitochondrial staining against this hazy background. So in the interest of better defining our mitochondria to generate our mitochondrial mask, we're going to use a Modify Image tool. This menu contains a series of filters that allow us to highlight our objects of interest in our grayscale images before we generate a segmentation of those images. So the tool that we've chosen from this menu is called Top Hat. This basically allows us to highlight the brightest pixels within a 5-pixel range. So think of it as like a paintbrush. And when we apply that filter, and we make this result image a little bit brighter here, you'll see that the filter excludes the hazy background. So this is going to allow us, when we try and segment our mitochondria, when we use the software to detect our mitochondrial network, it's going to allow us to better define those mitochondria. So as I mentioned in the strategy portion of the talk, we're looking to define both fragmented and fibrous mitochondrial networks. To define our fragmented mitochondria, the first step is to define objects that are small and round. So we're using a tool from the Find Objects menu here called Find Round Objects. This is going to bias our software towards finding small round objects. And when I click Apply, we can see that it's actually picking up quite a few objects as round. Some of these will overlay fibrous mitochondria, but we have a way of excluding those. So what we want to do is ultimately include in our final fragmented mitochondria result mask only mitochondria that are fragmented and discrete from the fibrous mitochondria. So the next step in building this analysis is finding our fibrous mitochondria. So again, from the Find Objects panel of tools, we're going to select this time the Find Fibers tool. And we're going to choose again our refined mitochondria, so the result image of our Top Hat filter. And in this case, we're going to aim to select the fibrous networks of our mitochondria. So we're defining these by the minimum and maximum width of our fibers as well as the intensity above local background. So again, very similar to the input for a standard application module. And what we're interested in here, if we look at the side-by-side analyses, is actually identifying, using the mask that's generated from the fibrous object. And that's going to be what we define as our fibrous mitochondrial mask, as you see defined here. Now, as I mentioned, we want to ensure that our fragmented mitochondria are discrete from our fibrous mitochondria. So to do that, we're going to use a tool from the Modify Objects menu called Remove Marked Objects. This is going to allow us to remove any fragmented mitochondria that overlay our fibrous mitochondrial mask. So now the result mask that we see here will contain only mitochondria that are fragmented. If we zoom out a bit, we can use, again, the split view tool that helps us confirm that any fragmented mitochondria that are coincident with our fibrous mitochondrial networks are now excluded. So this is the result that we want from measuring our fragmented mitochondria. We also want to make measurements on the total mitochondrial network. So what we want to do here is combine our fragmented mitochondrial mass with our fibrous mitochondria mass, again using a logical operations tool from the Modify Objects menu. And again, we're going to combine those two masks with an OR because we want all of the objects that are identified in both masks. You can see here when we review the side-by-side, that is the case. Now, we mentioned earlier that we want to define a peri-nuclear region and then make measurements of mitochondria within that peri-nuclear region. So to do this, what we're going to do is we're going to use the nuclear mass, and we're going to grow our nuclei by the number of pixels that we want to define our ring by. So in this case, we're looking to define a 15-pixel peri-nuclear ring. And we're going to use from the Modify Objects menu, again, the grow objects without touching. So this is going to allow us to grow our nuclei, but exclude regions where the nuclei are touching. So you can see here, if we apply a grow step with a 15-pixel output, we'll see that while these nuclei are adjacent, it will not encroach on a nucleus that is touching. So it's not going to grow past that limit, so you'll still be able to retain a robust segmentation of your nuclei. Now, once we've generated our fat nuclei, as I like to call them, we can then create our ring mass with a logical operation. So again, from that Modify Objects menu, we can take our fat nuclei and we can actually subtract out the positive nuclei that we identified in our earlier step in our cell scoring. And in this case, we're going to use the and-not function to combine them. So we're going to take the fat nuclei, subtract out the positive nuclei with an and-not, and this is going to give us our 15-pixel peri-nuclear mass. Now, the final step in building this segmentation is to then define the mitochondria that are in that peri-nuclear region. So what we can do then is we can do another logical operation for the Modify Objects menu. And in this case, we want to keep mitochondria that are within the peri-nuclear mass. So in this case, we're going to use an AND function, and this is going to give us a mass of mitochondria that are only coincident with that peri-nuclear region that we've defined, as you can see here. So side by side. Now our final step again is to generate our measurement set. In this case, we're going to use our whole cell mass again as our outermost boundary. We're going to make a series of measurements on our whole cells, everything from area to shape factor to the integrated intensity of our mitochondrial stain. And we're going to, again, make measurements of intensity, shape, area, etc. on our total mitochondrial mass, our fragmented mitochondrial mass, our fibrous mitochondrial mass, as well as our mitochondria coincident with the peri-nuclear region. And we also want to make some measurements on our nuclei because, as you're aware, oftentimes, especially in toxicity assays, you'll see an effect on nuclear morphology or intensity as toxicity increases with certain treatments. So then when we click Apply, we can review the result mask for this particular application module that we've generated. And we can see, as we zoom out here, that we're very nicely picking out, and we can toggle the map on and off, the boundaries of our cells, as well as our mitochondrial networks, our nuclei, and our peri-nuclear region. Now, keeping in mind that we are defining the peri-nuclear region here, and we're making measurements on it, but all of those fibrous and fragmented mitochondria within that region are still being counted as in their individual classes as well. And again, you can see in the data table here on the right that we can review all of our data points. We can actually sort them by clicking on the column header. So if you want to define a range of those, we can. And again, once we're happy with our result mask, we can save that module and immediately run it on everything from a single site to a stack of plates. This next slide here shows you a preview of the data that we've generated with this particular module. You can see that as we predicted from looking at our image montage, we do see a significant impact of some of these treatments as we go up the concentration range on the average cell count. So we're actually losing quite a bit of cells in this range. The mitochondrial shape factor is changing with some of these treatments. As we move closer to 1, these mitochondria are becoming more round. So that's similar to the effect that we saw in the publication that we highlighted earlier in the slides. Additionally, we're seeing an impact on the integrated intensity of our peri-nuclear mitochondria. So in this particular case, before we see acute toxicity, we're actually seeing a modest increase in our peri-nuclear mitochondrial intensity, which then goes down as we begin to lose cells, which is what you would predict. So this is just a snapshot of some of the many interesting outputs that we can generate from, again, a very straightforward custom module that's based on largely upon our application modules, and then we just build on the capability of those application modules to provide a simple way to access a much more complex biology. So I hope that these examples have proven useful to you to see the power of our custom module editor. Again, it's a great tool, bridging the capabilities of our plug-and-play turnkey application module and allowing you easy access to the power of our journaling interface, providing you with a comprehensive package of image analysis tools that allow you to access complex biologies in a very simple manner. So thanks again for your time today, and we're more than happy to take any questions you may have. And with that, I'll turn it back over to Grisha.
Vicki, thanks so much for going through all that. That's quite a number of parameters you're pulling out of a couple of colors there of a biological assay. At this time, I'll just go back to discussing the hardware that those images can be acquired on. Vicki talked a little bit about MetaXpress, and then think about your questions to submit by the Q&A window for Vicki. So Vicki talked about MetaXpress and the new analysis capabilities in MetaXpress to generate custom modules. I'm going to talk a little bit about, have a handful of slides on the MetaXpress Micro XLS. So it's building on our wide field automated imager. Continue to configure it for your needs with up to 5 filter cubes at once, and of course order more cubes. You can order the cubes as single or multi-pass filters. Variety of light source options are available to meet your application needs. 1 to 100x air and oil objectives. Camera options continue to offer the standard with the CCD sensor and the SC mode for the large field of view and high dynamic range imaging. Digital confocal option, and one of the biggest improvements to the XLS right now is to increase the speed. You can go up to 50,000 wells a day, and what does a well look like? That's a low resolution 10x objective imaging a whole 1536 oil plate, or a 4x objective imaging a single well of a 3D4 all-in-one image that comes from that large field of view and high dynamic range sensor. The XLS is still available with the transmitted light option, the fluidics, and world-class environmental control option for temperature, humidity, and CO2 control, and that allows for these multi-day.
Sorry to interrupt.
Yes.
I think we can't see your slides.
Thank you.
Yeah, sorry about that.
I apologize for those online. I will whip through those again. See them now? Vicki?
1 second. We can see. Yeah.
You can see them now?
Yes.
Okay. So sorry about that. So you heard what I was mentioning. Vicky talked about MetaXpress. I'm going to talk a little bit about the ImageXpress Micro. So the XLR system is still configured for your needs. 5 filter cubes at once. Cubes are available as single or multi-pass. Multiple light source options to fit your application needs. Variety of objectives. We have the CCD, the standard system, as well as the FCMOS with a large field of view. And that field of view allows, for example, with a 10x objective to capture a whole 1536 well in a single shot, or at 4x a whole 384 well in a single shot for low resolution imaging. Still available with transient light, fluidics, environment control options, and the environment control controls both temperature, humidity, and CO2. And here is a multi-day experiment where the cells are quite happy, so happy that they divide in the cell. And this image was not, this movie was not taken sitting in a single well, but going from well to well to well. And there's more movement of the cells than the system going on, so you can follow live cell events. Just to show you the field of view, that's a standard system. What the field of view would you get with our standard system, and not capturing the whole well with a 4x objective. With XLS, you can capture the whole well. Light source options are now available to address a variety of different applications. If you have dyes that need to be excited with UV, DIR, ratiometric imaging, or fast excitation switching. So the ImageXpress Micro is used for a wide variety of high-content imaging, all the way from whole animal to subcellular. Vicky concentrated on subcellular events in developing modules, but people do things with a wide range in between. And now the XLS has improved the speed at which you can acquire that data. That 50,000 wells a day comes from extrapolating out a 2-color assay that would have taken 15 minutes previously will now take 10 minutes. And actually, in hands of some of our beta customers, they're actually beating that speed with a 2-color assay. But in our hands, 2-color assay is taking 10 minutes. A 2-color 96-well plate will take 3 minutes, at least in our hands. So MetaXpress custom modules, which Vicky went through 2 interesting examples of quite complicated measurements that you want to extract from an assay, but we can do a variety of things: mitotic spindle, centrosome imaging, proliferation with transmitted light, nucleate outgrowth, and puncta. Those are little puncta within the nucleates or near the nucleates can be quantified, and migration assays. And what, as Vicky alluded to, once you create a module, whether it's a custom or a standard application module, those things can be run across plates, and we don't want analysis to be a bottleneck. So we have MetaXpress Power Core, which is an option to run each of those modules across a different CPU of a computer, and that will drive your analysis time to be faster than acquisition. So I've moved our bar down for how long acquisition time might take, and somewhere between 8 and 16 cores of Power Core, you can have that module run faster than acquisition to keep up with the acquisition. So ImageXpress Micro XLS cut acquisition time by up to 33%. Choose your own light source, create custom analysis modules. We didn't cover, but those modules can also be run over time-lapse data, so you can analyze that time-lapse data more easily now. Keep up with that. Keep your analysis going as faster than acquisition. And we also didn't cover accommodating a variety of image formats into the molecular devices workflow by importing those images. So on that note, I'll remind you about how to do the questions. If you have questions that you would like to ask after the fact, here's my email address, and we can also put you in touch with Vicky. One of the most common questions is, when will this webinar be posted? It will be posted within less than 1 week, on our web page where we host our webinars. You can look at past webinars there, methoddevices.com/ATS webinars. If you are an existing customer who would like to do a webinar to talk about your work, we welcome that at any time. These modules can be shared, and we have a forum where journals and modules can be shared with each other so that you do not need to recreate that. That software forum is methoddevices.com/forum. And you can interact with other users on that forum. So to remind you how to submit questions, please go to the top, click on the Q&A, type in a question, choose all panelists, press the send button. So let me pull up the Q&A. So there's a couple of questions. So Vicky, the example analysis you did with the Transfluor module, just looking for puncta in different compartments, what other applications or biological assays could that be used with?
Thank you, Risha. So it's actually a very flexible application. We can look at anything from bacterial engulfment in macrophage, for instance. We can look at lysosomal staining or lipid inclusions for other types of toxicity reads. We could create a multi-wavelength Transfluor type application. So you could look at multiple endpoints in a single assay, maybe translocation of a receptor in addition to tox readout. Additionally, if you wanted to restrict your puncta measurements to the nucleus, you could look at anything from methylation of DNA or histones to DNA damage markers. And it's just the custom module gives you, again, additional flexibility to build in those reads working from the very powerful Transfluor module that we've developed as a plug-and-play.
That's great. Let me just look at these questions again. In the mitochondrial example, and just as a reminder, your desktop is not shared if you're going to show something. In the mitochondrial example, why did you choose 15 pixels, and is that for the ring of the area around the mitochondria that you want to measure, and is that modifiable?
Actually, it is very easy to modify that ring. So I chose 15 pixels just as a starting point. It's very easy to create alternate versions of the module where you change the width of that ring, and then you can just run them across your data set and see which gives you the most relevant reads, in this case for your peri-nuclear mitochondrial staining. To make that change, we would actually just alter the grow step that we use to generate that ring mask. So we grew the nuclei by 15 pixels in that example. If we wanted to make it larger or smaller, we would just grow it by whatever amount that we considered relevant, and then perform the same subtraction step with the original nuclear mask. So this is how we would alter the width of that ring, and then again save the module as a different version and run it across your data set. So it's very easy to modify, just that one step.
Got it. What if you just, somebody's asking, what if you just want to measure something else about the mitochondria, like the localized signal or the intensity? Did you review that?
Sure. So that's also very easy to enable. Because we generated the ring mask, which we then combined with the mitochondrial mask to create that specific localized mitochondrial staining measurement in that peri-nuclear region. In this case, what we would do is, rather than generating that final result, we would simply use the ring mask and enable that as an object to measure within our cells. So in that measurement step tab at the end of the module, we would just define that ring as a region to measure, and then we could enable any number of measurements in that. So that it's an integrated or an average intensity of whatever stain we're interested in.
Okay. I think there's one last question, unless, and that is, and I'll take it. The question is, can you run these modules over time-lapse data? And the answer to that is yes. With MetaXpress 5.1, you can once a module is created, you can run that that module automatically over the time-lapse data, and the time-lapse data, the analysis is also compatible with MetaXpress PowerCore. On that note, if there are other questions, you're happy to email me. I will just hold it open for one more minute till the hour exactly to see if there's any other questions. I apologize for the waffles you're seeing. I'm just looking to see if there are any other questions showing up. Okay, thank you very much. We will keep you informed about future webinars. If one of you is a customer that has something that is that you think others would be interested in hearing about, your science and your biology, we'd be happy to host a webinar. And you can look at, I'll go back one slide to these go back one slide. So the webinar will be posted within 1 week to www.melassadevices.com ATS webinars. You can look at past webinars there, and we have some customer webinars posted as well. You can share modules with others on our forum, and in general, our information on our products can be found at www.melassadevices.com/ATS.